Welcome to what really is our last fully vodcasted lecture. I promise this is for real. So it's a couple of announcements that I want to make today just in terms of the post-test knowledge survey that I know many of you have already taken time to fill out. But if you haven't had a chance yet to do that, please, I ask for your help with that. It really is such an immense learning tool for me, and I think it also helps you see what you have learned. And of course, remember that it's also worth 5 points of extra credit on the final exam, as well as a replacement of your low homework score. So in terms of the poster presentations, things continue to roll along. Please feel free to continue to submit drafts of your poster through Friday for feedback. And please do make sure that you've thought of some way to print your posters, or you have a plan in action for that, so that you're not having to wait till the last minute. The final exam that you're all probably already working on is due on Monday, April the 30th. So if you're watching this vodcast at the regularly scheduled lecture time, that means a week from today we'll be meeting in the ASMB foyer, just right outside where the classroom is, and we'll be setting up our poster boards out there, and by those, those little water fountains that are out as you come in the front doors, and we'll be going through those presentations, and they're totally chill, totally laid back. But the way that we're going to do those is that half of the class will be the first to present. Half of the class will present from 10:15 to 11:15. And what that means is that you'll stand by your poster and you'll say, here's what we did, and you'll get engaged with discussions with people that might visit your poster. And then when we hit 11:15, I'll basically just say, okay, let's switch so that we can have the other half of the group become the presenters, and those that we're presenting will become the consumers of all of that great information, and you'll head around and help to assess people's posters. And actually, that's a great time to mention that you will be grading your group members. You will also be assessing the posters that you go to visit, so you'll give them a score on the quality of their presentation and their poster. And this is so very important, because really this is peer assessment in action, and that's the cornerstone of science. At some point in science, you come to the place where you no longer have a professor who gives you feedback, and instead we rely on each other to do so. So it's so important to give reflective feedback of your peers and to really take that peer assessment seriously. Look at the poster, listen to the presentation, and think about the work that went into that so that you can acknowledge those groups that put in the most effort. It's sometimes important to show them that they are the ones that did a really good job, and by comparison, maybe there are some others that weren't just quite as good. So find a rating. We'll be rating them on a scale of 1 to 5, the quality of those presentations. And please don't feel as though you have to visit every single poster out there. Truly, you do not need to do that. You can go around to a couple of posters that you're the most interested in, and then become super engaged with the presenters at those posters, and maybe spend 10-15 minutes at the poster that you're really enjoying. And just make sure to give feedback to those poster groups who you do attend. In fact, I put less stock in the feedback of people who seem to have assessed every poster. Not likely, right? Not likely that you actually made it around to every single poster out there. So, in fact, I really would prefer that you just give thoughtful feedback to those posters that you do visit. So once again, if you are watching this at the regular, lecture time, tomorrow is our second practical exam in lab. So be ready for that. Brush up on some of your knowledge from the lab stuff so that you can come in kicking butt and ready to go on that lab exam. Sweet. Let's go ahead and jump in, because when we left off, I was literally totally jazzed on the cyanobacteria, and we'd been talking about how diverse they are, not only morphologically, but also with respect to reproduction strategies, as well as their ability to live in diverse environments. We had looked at how they can live in hot spring type environments, but also in just regular lake water or in soils. And so today we're following up on some of that, and I want to begin with a really cool picture that I took when I was biking from Fort Collins to Denver, and I was kind of going the back way, kind of near Estes Park. And what you can see here is a cool picture of what happens when cyanobacteria grow up to too high of a concentration. This is something that is termed a harmful algal bloom, even though, Don't get me wrong here. Cyanobacteria are not algae, but for many years they were thought to be algae, and so they're even called, sometimes if you see them on food labels and whatnot, they're called blue-green algae, and I'm going to jot that down. They are still known as blue-green algae, even though once again, that is incorrect. They are not algae, they are cyanobacteria, but old terms die hard. And so this is called, what we're seeing here is called an HAB, or harmful algal bloom. And these occur when the concentrations of nutrients become too high in the water. And by those nutrients, I'm referring largely to NPK, nitrogen, phosphorus, and potassium, that can be from excess runoff of fertilizers, for example, into the environment, giving rise to these overgrowths of algae, in this case cyanobacteria. And so what can happen is, this was kind of midsummer when I took this photograph, but if I had come back by later in the summer, what we would see is that the cyanobacterial bloom would die off, and when it dies, it will form something called a nuisance bloom. And this nuisance bloom will get really smelly and icky, and you kind of stick your hand in and pull up a handful of scum, and this nuisance bloom really can be truly a nuisance. So let's jot down that excessive growth causes these surface mats, or these blooms, and in the summer when it hits the warmest months and it dies off, it creates this nuisance bloom, and it really can be quite a nuisance. It can be quite a problem. And so again, I know this interests Coulter, but this is something we need to think about environmentally, in terms of allowing excess nutrients to run off into the water. And I'm going to say this more than one time today, so I guess maybe my ball chair here in my office, this is kind of like my soapbox today, so forgive me. All right, let's talk about another environment, and I don't know, this may be even cool. No, this is cooler. This is definitely cooler. Let's talk about how cyanobacteria can grow in even the most desertous of soils. So every environmental niche is typified differently by cyanobacteria: freshwater, marine habitats, soils, but also rock surfaces, and even, yes, desertous soils. So if any of you are like me and you like to spend a certain amount of time each spring in Utah and go to the Canyonlands in Utah. I'm taking my ski team backpacking there the week after finals week. So this is a cool place to see some of the magic of cyanobacteria, and it truly is magical to see how the cyanobacteria can grow in these long trichomes, like we looked at the picture of the other day, and they secrete this mucilaginous ooze that allows them to gradually tie up the particles of soil in the mucilage that they secrete, and it eventually gives rise to a visible structure called a microbiotic or a cryptobiotic crust. And this is a picture that I took in 2004 in Utah on the above the canyons outside of Lake Powell, outside of particularly a canyon named Moki Canyon. And you can see that it's grown to a large size. This crust is maybe a couple of inches tall. And I know that doesn't seem particularly wondrous, but when you realize that it literally takes the cyanobacteria up to 200 years to grow this knob, it gives a little bit more meaning to the size of that. And so as these crusts grow, the cyanobacteria do a number of things for the soil. They obviously, because of their outer sort of mucilaginous polypeptide regions, they can take on more water, and as they take on more water, they allow the soil to retain the water better, and they also forming these knobs, they help prevent erosion. So I'm actually going to jot some of this down, some of the benefits of the cyanobacteria in these microbiotic crusts. One is that they increase water retention. And two, we can say that they're also so very important in fighting erosion. And I'm going to leave the last one for the next set of pictures that I have, because we'll see in the next shot a place where the microbiotic crust has grown up to very large sizes. And you'll notice that a big part of the reason that it has been able to grow like that is because it's protected by a log that has been keeping the foot traffic away from the crust. So we see in this picture that really nice, well-developed, mature soil crust. But out here you can see where this is actually in BLM land, so it's actually free range for cattle. And you can see where they've stomped down the crust in these areas. So we recognize that if it's allowed to grow to these higher these larger knobs, then it can actually fix a lot of nitrogen, right? And enrich the soil. So that's the third thing that you want to write down about your cryptobiotic crusts, is that number 3, they fix nitrogen, and in doing so, they enrich the soil. And in enriching the soil, they pave the way for other things to grow, such as this awesome looking desert wildflower. Looks like a penstemon to me. So pretty cool how cyanobacteria can pave the way for other life. So today you might notice that I am in fact wearing my Don't Bust the Crust badge. This is a badge that one of my students brought back for me, and let me see if I can put it up a little closer, where you can see the shoe coming down on the 200-year-old cryptobiotic crust. So please don't bust the crust, and if you do go visit Southeast Utah or the Colorado area where in the canyon lands there outside of Grand Junction, definitely don't bust the crust. And I've got a picture here that I'll bring up as well. And I've also got some handouts, some leaflets that come from some of the places in Utah. This one is a handout from the USGS, and it talks a little bit about the impacts of the soil cyanobacteria on enriching the soil, but it also talks a bit about human impacts and how to help stop the human impacts on the soil. So some good stuff in this particular article about the microbiotic crust. And I also have one other one that is from Canyonlands as well in Southeast Utah, and you can see in here it kind of goes through and summarizes the 3 things that the crust really does for us, and in terms of increasing water retention, decreasing soil erosion, and also fixing nitrogen and really enriching the soil. So you can see in this one picture on this the way in which the trichomes and filamentous structures of the cyanobacteria really wrap up the soil particles and help hold on to those soil particles. So pretty amazing job that the cyanobacteria do as a part of the crust. Sweet. Okay, if you really ever want to wear my badge, I'll let you. Let's take this opportunity to revisit our Winogradsky column, and I want to make a couple of quick notes, make sure that everybody's gotten the labeling for the Winogradsky column. And there's a nice picture here that you can add to your notes if you haven't already done something like this. Remembering that deep down in the Winogradsky column are our organisms that live anaerobically. In that anaerobic sediment, we see things like Clostridium and Desulfovibrio are anaerobically respiring and are fermentative organisms. Of course, the things stay anaerobic at the interface between the anaerobic soil and the anaerobic mud, and we see that being the place where the green sulfur bacteria as well as the purple sulfur bacteria are growing and living. In terms of the purple sulfur bacteria, things like Thiospirillum and Chromatium. In terms of the green sulfur bacteria, things like Chlorobium and Peladictium. And then, of course, as we move our way up, remember so well developed in Cody's column were the purple non-sulfur bacteria and things like Rhodopseudomonas, Rhodocyclus, and whatnot that were very, very much a part of that sort of wayfishly looking purple in the more anaerobic water, but in reaching up even into the more aerobic water, because remember their flexibility metabolically. And then at the very top we had our aerobic regions where the cyanobacteria such as nostoc as well as Synococcus were making their home. So hopefully that helps kind of summarize the Winogradsky, though I do want to add to this just a little bit. I want to talk just a moment about some other bacteria that may live in the Winogradsky, though we'll think about them also in their broader environmental roles. So we'll begin with the aerobic chemoautotrophs, including the nitrifiers, which the nitrifiers, as you well have guessed, play a huge role in the nitrogen cycle. And of course, so does cyanobacterium. So the other day I brought in my little chart that Emily had made that shows the nitrogen cycle, and I'm going to grab that too here.